Hi, class. This is the Module 10 lecture. The topic is energy and cellular respiration. So we're going to spend today's time learning about cellular respiration. And this will complete our study or complete the formal study of energy that has spanned several of our modules lately. We've got one more module to go that will wrap up the Unit 2 information. So we're getting through the semester. So before we begin, let's remind ourselves, please, of the balanced chemical equation for cellular respiration. It begins with a molecule of glucose. It is possible that other monosaccharides can be fed into cellular respiration. So I don't want you to think that it only begins with glucose. We're going to study it, though, beginning with glucose because it's a common source of starting material for cellular respiration. So it begins with one molecule of glucose. Glucose has 6 carbons, 12 hydrogens, and 6 oxygens. One molecule of glucose reacts with 6 molecules of molecular oxygen, meaning that there are 2 oxygens bound together, 6 of those, through all 3 stages of cellular respiration. We will learn those 3 stages today. 6 molecules of carbon dioxide are produced, 6 molecules of water, plus energy in the form of ATP. I mean, after all, this is the purpose of cellular respiration, to generate energy out of the molecules that we consume in our food. Some of the energy is going to be released to the environment in the form of heat, which for mammals like ourselves that are endothermic, we actually utilize that heat. We utilize it to drive part of our core body temperature. So yes, it's wasted from the reaction of cellular respiration, but it's utilized. We have evolved to utilize that heat, that thermal energy. Remember that the first law of thermodynamics states that the energy in the universe is constant, that it changes forms. And I think this is a really good example of that at the level of the organism like ourselves, and also at the level of the molecules that are going through this chemical equation. We have some released in the form of heat, but that energy is being used for another purpose. So we're pretty efficient at utilizing the energy. All right, so we did see this slide in Module 9. This is a summary representation of the energy hill, if you call it that. Imagine that we have two axes. On our x-axis is time or progress of the reaction. On the y-axis would be increasing energy. So the higher we are, the higher the energy. So the process begins, this process of the electron transport chain, which you've probably heard of. It begins when the NAD plus NAD, the oxidized form of NAD, is loaded with electrons. And this happens during stages 2 and 3 of cellular respiration. So these molecules come in loaded with their electrons, and then the electrons go through a series of different proteins that make up the electron transport chain. So our electrons are going through the electron transport chain. Our protons, though, are also present. Remember that every element has electrons, protons, and neutrons. The electron transport chain describes the events for only the electrons. And that's why I actually don't use the name electron transport chain for the 3rd stage of cellular respiration, because it implies that there's only a role for the electrons. And there is. But there's also a very important role for the protons that are coming through. remember that every molecule of hydrogen, which is what we're talking about with our electron transfer, every molecule of hydrogen has one electron, one proton, and one neutron. So those protons have a very important role too. The way I think of the shuttles, these energy shuttles or electron carriers, is that they sit empty, like we're seeing here in NAD+. But when they are reduced, reduction involves gain of electrons, we see a nice place for the electrons. But what we have to remember is that the protons of hydrogen are riding along too. So the way I think about it is that NAD+ is the carrier, the electrons sit in the seat, and then they're holding hands, if you will, with their proton still. They're helping to bring the proton through as well. And the proton is really important in generating ATP. In fact, most of the energy that comes out of aerobic cellular respiration ties into these protons. Now, as our electrons are shuttled through the electron transport chain, as we move down in our figure, the energy is decreasing. So another way of thinking about that is the higher we start, and we being an electron, just pretend like you're hydrogen. That's what we are. The higher we start as hydrogen going into the electron shuttle NAD+, the higher energy we are, the more space there is to fall in a controlled way. Now, at the very bottom of the energy hill is a very important molecule, 1/2 of O2, 1/2 of molecular oxygen. It is incorrect to simply say oxygen. Oxygen, 1O, does not exist in nature. It's too unstable. I mean, that's the oxygen free radical. So instead, we need to describe it correctly based on its chemistry. Let's look at this another way. Let me get a text box going. So if we have a molecule of O2 and we react it with 2 hydrogens, what do we get? We would get H2O2. That is not water, that is hydrogen peroxide. However, if we begin with 1/2 of O2 and react that with 2 hydrogens, now we form water. Oops, sorry, wrong. Oh, there we go. So 1/2 of O2 with 2 hydrogens becomes H2O. Okay, so there's a big difference here. It's important that we acknowledge that at the bottom of the energy hill is 1/2 of molecular oxygen, 1O, so that when those 2 hydrogens come together with the 1O, it creates a molecule of water. All right, let me get rid of my text box. That's always tricky with Zoom, and I don't know why. So just bear with me a moment. I've got to try to get rid of it without being able to see my mouse prompt. Okay, this isn't working so well. There we go. Okay, now we can continue. There we go. So again, it's 1/2 of O2 that's at the bottom of the energy hill, which produces water. You know that water is one of the products of cellular respiration. It is produced during stage 3. It's not until stage 3 that water is produced. So when we're going through these different stages of cellular respiration, let me back us up. When we go through the 3 stages of cellular respiration, I would like you to think about what material we start with, what material we end with, but also what material ending in stage 1 goes to stage 2. What material do we end in stage 2 that goes to stage 3? So you'll see that the homework assignment for module 10 is a table that you're going to fill out. And yes, most of the answers are right here in the lecture. So you could just basically copy the answers out of the tables that are in this slide set. But please take the time to understand what it means. Take the time to actually learn it. Hopefully I present it in a way that makes sense to you. So we have 3 stages of cellular respiration. The further that we get through, the more energy that is produced. So let's go through these stages. Just something to think about are the chemical characteristics of oxygen that facilitate the creation of water. In other words, what is it about oxygen that attracts those protons from hydrogen to it to make water? I'll give you a little hint. Look at your periodic table. Look how close oxygen is to fluorine. It's right next door. The answer is the electronegativity of oxygen. Oxygen is far more electronegative than is hydrogen. So the attraction of those electrons to go to oxygen, also pulling those protons through, is what gives us our chemical characteristics that define cellular respiration. Oxygen is electronegative. It loves electrons. All right, so we have 3 stages in cellular respiration. We're gonna just briefly, I'm gonna give you an overview of the 3 stages, and then we're gonna dig deeper. We're gonna dig into each of the 3 stages. At the end of the lecture, I am gonna play a YouTube video for you. It's one of the YouTube videos I put up in your supplemental video list for Mod 10. But I'm going to play it here because I think it's a really important video in helping to explain some of the important concepts of cellular respiration. So that's what my way of hopefully getting you to watch that video. So stage 1 is called glycolysis. There is a transition step that happens between stage 1 and stage 2. If this transition step does not occur, we do not progress to stage 2. What happens here in the transition step is that the product of glycolysis, which is called pyruvate, is oxidized. So don't worry about getting all these terms written down now. We are going to break them down stage by stage. Right now, we're just having an overview so that we can kind of get the big picture. So stage 1 is glycolysis. It begins with 1 molecule of glucose going into glycolysis. We complete glycolysis, followed by the transition step, and then it moves to what is called the citric acid cycle, TCA. It's also called the Krebs cycle after Charlie Krebs, who discovered it, which, by the way, yes, I'm a nerd. Charlie Krebs was a local hero. He's from the San Diego area. In fact, his grandson is a physician, a house physician within the Scripps medical system right here in San Diego. I got to meet him. It was really cool. And yes, I'm a nerd, and I'm proud of that. Stage 3 is the electron transport system. There is a process, or it happens through what is called oxidative phosphorylation. We've learned about substrate phosphorylation so far when we talked about enzymes. This is a different process. It is a different way of adding a phosphate group on to create ATP using oxygen in the environment. There is a role of the electrons. They go through the electron transport chain. But there is also a very important role for each of those protons called the proton motive force, which is also known as chemiosmosis. The person who discovered chemiosmosis did win the Nobel Prize in recognition for these wonderful contributions to biology, to all of life. Okay, so let's go through glycolysis. It occurs in the cytosol of the cell. The mitochondria is the organelle in which cellular respiration occurs. That's true. But glycolysis happens in the cytosol. It does not require a specific part of the mitochondria in order for that to happen. It's happening outside of the mitochondria. It is the beginning step of cellular respiration. This is true for both aerobic and anaerobic cellular respiration. Glycolysis is in both pathways, aerobic and anaerobic. And what it does is it takes that single molecule of glucose or whatever monosaccharide we're beginning with, and it breaks it down first. So if we begin with glucose, which has 6 carbons, it breaks it down into 2 of the same molecules called pyruvate. Each pyruvate has 3 carbons. Glucose has 6 carbons. So that means, due to the laws of thermodynamics, that energy or matter isn't created or destroyed. It just changes forms. That means that we have to have 2 molecules of pyruvate to account for the 6 carbons that are present in glucose. So again, glycolysis is used for both aerobic in the presence of oxygen and anaerobic in the absence of oxygen respiration. We will spend a little bit of time at the end of this module talking about the different types of anaerobes. What I would like you to know now is that anaerobic means that it doesn't utilize oxygen in the chemical reaction. It doesn't mean that it's not there. It could be present in the environment, but when we're talking about an anaerobic stage of glycolysis, it just means that there's not oxygen being used in those specific chemical reactions. There's a difference between oxygen being used versus oxygen being present in the environment. The transition step is next, and it takes those 2 molecules of pyruvate and oxidizes them. It changes pyruvate into the starting material for stage 2. Okay, so pyruvate is the ending material of glycolysis, but not the starting material of the citric acid cycle. It's modified, it's oxidized. Now, as we begin stage 2 and stage 3 for that matter, we are now moving in to the mitochondria, the organelle associated with respiration in eukaryotes. Specifically, we're in the inner mitochondrial membrane. That is where the cellular machinery that allows the electron transport chain to happen, that is where it is located, right in that inner mitochondrial membrane, which is also referred to as the cristae of the mitochondria. Location is very important, not just in real estate, but also in biology, right? You have to have your materials in the right place so that the chemical reaction can occur. So in stage 2, the oxidized pyruvate is converted into another compound that contains 2 carbons. So we begin stage 2 with 3 carbon molecules, 2 of them, and then we end it with 2 carbon molecules. What's one of the products of cellular respiration? Carbon dioxide. And carbon dioxide has a carbon. Let's see where that is produced. The importance of the citric acid cycle really is not in generating energy. It only produces 2 molecules of ATP. It's more to supply the 3rd stage with the electrons and the riding along protons that go with those electron shuttles. So stage 2 begins with the oxidized pyruvate. I'll show you the name of that in just a moment. But it ends with our electron carriers in reduced form, loaded up with electrons so that they may go through stage 3, the electron transport system. Only a tiny bit of energy is made during glycolysis and also during the citric acid cycle. For 1 molecule of glucose, glycolysis only produces a net production of 2 ATP. When we go through the citric acid cycle, another 2 ATP are produced, 1 for each molecule of oxidized pyruvate. So we've only produced a total of 4, a net production of 4 ATP molecules for 1 glucose going through stage 2. Now, stage 3 is where we get the big bang for the buck in terms of ATP. It occurs through the process called oxidative phosphorylation. We are creating ATP through the process of oxidative phosphorylation. This is different from substrate level phosphorylation. Stages 1 and 2 use substrate level phosphorylation. It's not until we get to stage 3 that we see the oxidative phosphorylation. And this means oxygen is used. So even though oxygen is present in the environment during stage 2, it is not used in the chemical reactions of the citric acid cycle. It's just present in the environment. That does make it aerobic, but it's not aerobic because it's using oxygen. It is aerobic because oxygen is present in the environment. Stage 3 is where the oxygen is used. And you'll see exactly what it's used for. It's used to make water. So our electron shuttles, the oxidized forms, that's what the plus means, there's no electrons there, NAD and FAD, they both work as carriers to shuttle electrons into the electron transport chain. This is occurring in that inner mitochondrial membrane, meaning that the cellular machinery, the electron transport chain itself, is located in that inner mitochondrial membrane. And you may recall that the inner membrane is all wiggly and wavy. The outer membrane isn't. That is an evolutionary adaptation to increase the surface area to volume ratio of the cell or of this organelle, the mitochondrial. So that means that there's a lot more machinery for cellular respiration since there is more inner mitochondrial membrane due to all those wiggles. So this is where we get the most ATP produced. Our electrons are finally passed through the chain to the terminal electron acceptor or the final electron acceptor. Both terms are correct, which is 1/2 of molecular oxygen. It is not correct to say O2. It is not correct to say O. It must be 1/2 of molecular oxygen because that is what exists in nature. Let me show you the chemical reactions that support this as we go through. So we have the role of the electrons that go through the electron transport chain, but then we have that proton motive force, which has been discovered more recently than the rest of cellular respiration. When I learned about cellular respiration, I graduated from undergrad in 1997. I didn't learn anything about chemiosmosis. It wasn't worked out then. I learned about the electron transport chain, but now we know that there is a big role for the proton motive force, otherwise known as chemiosmosis. Okay, so in that stage 3, again, I'm emphasizing this, I'm being redundant on purpose. We have the role of the electron transport chain for the electrons. But we also have a pathway for the protons. They go through a molecule called ATP synthase. Well, let's just take a minute and look at that word, please. The ASE ending tells us it's an enzyme that is synthesizing ATP. So ATP synthase is an enzyme a pathway to make ATP. And it does it by pumping the protons through. Okay, so we end up with a concentration gradient of protons that is different across the membrane. I'll show you in a figure what I mean by this. But you know now that diffusion relies on a concentration gradient, and the feeding of protons into this molecule ATP synthase occurs because there is a proton gradient established. So we're using that potential energy of the concentration gradient to make ATP, right? We're not using energy, direct energy to make ATP. We're not harnessing ATP to make more ATP. We're using that gradient to drive the synthesis of ATP. Okay, so looking at the overview before digging into each stage, glycolysis begins with glucose, and it makes 2 molecules of pyruvate. Each pyruvate has a total of 3 carbons, but glucose has 6. That is why there are 2 arrows shown in glycolysis to indicate that there are 2 molecules of pyruvate. I would much rather you just put the 2 in front of pyruvate, by the way, if you're asked to draw this on your exam. We make a modest amount of ATP, only a net production of 2 for every 1 glucose. We are in the cytosol. We have not yet entered the mitochondria. Location is important. But beginning with the transition step, which is the oxidation of each of those 2 pyruvate, we now do require that inner membrane of the mitochondria. So glycolysis occurs in the cytosol. Everything else occurs in the inner mitochondrial membrane, where those cristae are. So pyruvate is oxidized. Pyruvate has 3 carbons, but the beginning of the citric acid cycle has 2 carbons. Yes, a molecule of carbon dioxide is produced in between the transition step, as well as during the citric acid cycle. And that allows us to account for all of our carbons. Now, as this oxidized pyruvate feeds into the citric acid cycle, we'll learn that name, the electrons are loaded into NAD+ and FAD+. So the ending product of the citric acid cycle that feeds to stage 3 are these loaded electron carriers. There is a chemical that actually ends the cycle. That's true. I want you to focus on what material feeds to the next stage, because that is what is relevant for cellular respiration and the generation of ATP. So if you were in chemistry, they may require you to learn all of these molecules. I'm not requiring that. I want you to understand what it means and apply that knowledge to a real world situation. The citric acid cycle produces another 2 molecules total of ATP. So far, with stage 1, glycolysis, and stage 2, citric acid cycle, there have been a total of 4 net molecules made of ATP. The loaded electron shuttles carry the electrons along with their riding protons. They're there too. It carries them into stage 3, which is the electron transport system. This happens by oxidative phosphorylation because oxygen is now utilized in the chemical reactions. Oxygen is present during citric acid cycle too, but it's not utilized. It's not rearranged into a different chemical molecule. That happens here. It happens by taking 1/2 of molecular oxygen. Your book shows this incorrectly. It should be 1/2 of O2, and reacting it with 2 protons to make a molecule of water. So oxygen is utilized during stage 3 in the production of water. Oxygen is reduced into water when it picks up those two protons and electrons. Now, by far, the majority of the ATP that is produced during aerobic cellular respiration happens here. I wish this ATP star were way bigger, proportionally related to the size of the ATP star for each of the first two stages. We're making way more here. We're making 28 to 32 molecules of ATP just in this one stage of the electron transport system. So by far the majority of it happens here. So that's the overview. We're now going to dig a little bit deeper and look at each of these stages. And again, please follow the carbons and follow the hydrogens. So what happens specifically in glycolysis? We begin with glucose, and it is cut by substrate level phosphorylation in half. It happens over several steps. Each of these stages has many chemical steps. We are going to concern ourselves with the overview of the entire stage. So glycolysis begins with one glucose, and it produces 2 molecules of pyruvate through substrate phosphorylation. This happens in the cytosol outside of the mitochondria. 2 molecules of NAD plus are loaded with electrons and they become reduced to NADH. So glycolysis also loads the electron carriers so that they may feed into the transition step. So we've got really two outcomes of glycolysis. Yes, pyruvate is produced. That's true, 2 molecules of it. But we also have loaded shuttles, reduced shuttles. We make only a net gain of 2 molecules of ATP. During glycolysis, a total of 4 ATP are made, but 2 of them are used. So that only leaves us with 2. Okay, so this is the summary of the chemical reaction of cellular respiration. Stage 1, glycolysis. So we're just at this stage. It begins with a 6-carbon glucose, 1, 2, 3, 4, 5, 6. It is enzymatically cut into 2 molecules of pyruvate. Each of those contains 3 carbons. So we do have our 6 carbons at the end, just like we started with. 2 molecules of adenine diphosphate are phosphorylated by substrate level phosphorylation to make a net production of 2 ATP. We do reduce 2 molecules of NAD plus into NADH during glycolysis. And then these electrons are going to be shuttled over into the citric acid cycle and over to the electron transport system. So let's take a look at that substrate level phosphorylation again. So we have a molecule of adenine diphosphate, and then we have the substrate, right? The substrate binds into the active site of the enzyme here. When that happens, this phosphate group attached to the end of this 3-carbon molecule is cut off, and it is added to adenine diphosphate to make ATP. So this is an example of substrate level phosphorylation. The enzyme physically holds the starting material in place so that cleavage can happen. All right, so that's glycolysis. We're now going to move through the transition step. The ending product of glycolysis is 2 molecules of pyruvate. However, the citric acid cycle does not start with pyruvate. The pyruvate undergoes this transition where it is oxidized. So the carboxyl group is removed and we produce carbon dioxide. We go from a 3-carbon molecule at the end of glycolysis to a 2-carbon molecule plus carbon dioxide. So we still have our 3 carbons. So this 2 carbon molecule that's left is oxidized. And to do that, oxidation requires reduction. Those reactions always occur together, the redox reactions. Because if a molecule is oxidized, something is reduced, right? That electron has to go somewhere. Oxidation involves loss. Where does it go? Reduction involves gain. It goes on to NAD plus and reduces it to NADH. There is an enzyme called coenzyme A that comes in and bonds with the remaining 2 carbons that are left. And now this is called acetyl coenzyme A, or acetyl-CoA for short. The citric acid cycle begins with 2 molecules of acetyl-CoA. That is the starting material, 2 molecules of acetyl-CoA. Okay, so let's look at that transition step. We're just looking at 1 pyruvate here, but we have 2, so it has to happen twice. Each pyruvate has 3 carbons. The first cleavage takes off one of the carbons and releases it in the form of carbon dioxide. So actually, we do need oxygen here to do that. And then we have the reduction of NAD plus that occurs when the electrons are loaded into it, reducing NAD plus into NADH. The protons are still there too. They don't just disappear. And then finally, this coenzyme A comes in and adds itself on to the 2 carbon molecule, creating acetyl-CoA, which is the starting material of the citric acid cycle. There is no energy produced during the transition step. All right, so now we're going to go to stage 2. It is a cycle, the citric acid cycle. So that means that the starting materials are fed through a series of chemical reactions that does produce an ending molecule here. We're not even going to discuss its name because it's not important for what we're learning. But that ending molecule feeds back into the cycle and keeps it going and going and going. Okay, so acetyl-CoA is the starting material for stage 2, the citric acid cycle. There are 2 of them for every 1 molecule of glucose. So that means this cycle turns 2 times. So you have to double the yields, right, to represent everything that happens from both molecules of pyruvate that is oxidized to acetyl-CoA. So first of all, we're going to lose the CoA, and that is released as carbon dioxide. 3 molecules of NAD plus are reduced into NADH. A molecule of adenine diphosphate is phosphorylated by substrate level phosphorylation to produce ATP. A single molecule of FAD is reduced to FADH2. That is the citric acid cycle. So the starting material is acetyl-CoA. The ending material that we are going to focus on is what goes to stage 3, the loaded NADH and FADH2 molecules. That is what goes to stage 3, our loaded electron carriers. So when we oxidize pyruvate, we get that acetyl-CoA, the carbon dioxide that is reduced, and then the reduced shuttles. So for each 1 turn of the citric acid cycle, we have 2 carbons that add together with CoA to make acetyl-CoA. 2 carbon dioxides are reduced, or excuse me, 2 carbon dioxide molecules are released, 1 from each pyruvate. And then we produce 3 molecules of NADH, 1 of FADH2, and a single ATP for each molecule of pyruvate. But we have 2 molecules of pyruvate, which means you have to double those. All right. So after 2 turns of the citric acid cycle, beginning with 1 molecule of glucose, our yields are 2 ATP, 6 NADHs, 2 FADH2s. All right, so here's a table that I've put together. And this is very similar to your homework assignment for Module 10, but you are asked to complete a table like this. So for glycolysis, citric acid cycle, and the electron transport system, glycolysis begins with glucose. It produces 2 molecules of pyruvate. A net production of 2 ATP are made. 2 molecules of NADH are prepared that then go through the rest of the process. The citric acid cycle begins with 2 molecules of acetyl-CoA. The ending material that is relevant for the next stage are those electrons and protons that are loaded on the reduced carriers. We do produce a net production of 2 ATP, 6 NADHs, and 2 FADH2s through the citric acid cycle when we turn it twice, 1 for each molecule of acetyl-CoA or oxidized pyruvate. So when I'm asking you to refer to the starting and ending materials, what I'm talking about is what passes to the next stage, please. That's what I'm looking for on your homework. That's also what I will be looking for on your exam. All right, our third stage occurs by oxidative phosphorylation to make ATP. So this does use the electron transport system, which includes the electron transport chain for the electrons, and then this process of chemiosmosis, or the proton motive force for the protons. We do need a good amount of oxygen here, because oxygen is the molecular oxygen is the final electron acceptor that picks up those protons to create water. So the electron carriers are what make this possible, and they are located in that inner mitochondrial membrane. We do generate a proton gradient across that inner mitochondrial membrane. As the electrons go into the electron transport chain, the protons are building up. They're left over. So we build up a gradient. And then we can use that concentration gradient to make ATP passively, right? A gradient means passive. It does not require energy. That would be active transport. So the electrons come out of our shuttles. They fall through the electron transport chain to where they get to the terminal or final electron acceptor, which is 1/2 of molecular oxygen. So oxygen picks up those hydrogens. It's electronegative. It loves those electrons that come with the hydrogen, and it creates water, which is one of the products of cellular respiration. So carbon dioxide is made in different stages, where the water is made here in stage 3. We're going to see that we generate a lot of energy by pumping those H pluses into the intermembrane space. So the H pluses, the proton gradient builds up in the intermembrane space. Let's look at why. That allows the protons to move through this molecule, ATP synthase, that synthesizes ATP. Okay, so here it is. So stage 3, we're looking at oxidative phosphorylation. So let's orient ourselves. So this cytosol is around. We have our phospholipid bilayer, which is the outer mitochondrial membrane shown up here. And then here's that inner mitochondrial membrane where all the wiggles are. And you can see that we've got some stuff here. So each of these big purple things are protein complexes. And we actually have 4 of them. So here's 1, here's 2, here's 3, and here's 4. this is the electron transport chain. So here comes our shuttle with lots of, with its loaded electrons. And what happens is NADH moves through this protein complex 1. These complexes are also called proton pumps. The reason why is the electrons come out and they continue through the chain, going through the subsequent parts of the electron transport chain. But the protons are pumped into this intermembrane space. So each of these protein complexes that makes up the electron transport chain is separating our hydrogen, the electrons and protons from hydrogen, if you will. So the protons are all building up in this inner membrane space. And we have a gradient. You can see that there are more protons in the inner membrane space than there are anywhere else. So the more that protons go through the whole chain, the more opportunity there is to generate ATP. So notice here that NADH feeds in before protein complex 1. FADH2 doesn't do that. It comes in after complex 2. So that means that we've lost the opportunity to harness energy out of the first and second complexes because FADH2 bypasses those first two. So depending upon which shuttles are loaded, there can be some variance in the amount of ATP produced. If we're using more NADH, if that's more readily available, the potential to generate more energy exists than if we're using FADH2 as our shuttle. So the protons go through each of these complexes, and then here's our descent to the terminal electron acceptor. 1/2 of O2 combines with 2 hydrogens, protons and electrons, right? The whole hydrogen, to make ATP. To make water. What's happening next, here's the production of ATP. All of these protons that are built up in the inner membrane space, they now move through this pump called ATP synthase that takes a proton, loads it into the rotor, and there's multiple positions in the rotor for multiple hydrogens. And then it pushes them through, turns the rotor, and phosphorylates a molecule of ADP by oxidative phosphorylation to make ATP. So this entire process is called oxidative phosphorylation since ATP is being made in the presence of oxygen. We're not looking at substrate level phosphorylation here, like we did with the first 2 stages. So chemiosmosis is where the most ATP is made, period. We make far more ATP through chemiosmosis than is made through any other process in aerobic cellular respiration. There are differences in the amount of ATP, the final count of ATP that are produced, depending upon the environment. So it's a range of ATP that is produced here, and things like how much energy is the cell needing to use at the time make a difference. If the cell is needing to use energy, it's not banking as much in the form of ATP. So we do see some variance in our quantities during stage 3. So here's a zoom in, a close-up view of the ATP synthase molecule. It takes the hydrogens, puts them in the rotor, and then cranks them. And as one of these H pluses goes through, we are phosphorylating a molecule of ADP to make ATP. So the yield of ATP that is made per proton going through ATP does change depending upon which organism it is and which carriers are being utilized. but it's close to a 1-to-1 on average for all eukaryotes, meaning one proton goes in, a molecule of ATP comes out. That is an approximation, and it depends upon the physical conditions and what the cell is doing at the time. So now let's fill in our table. Now we've got the information to complete stage 3, which begins with the electrons and protons that have been loaded onto the carriers, which are now reduced. The ending material is carbon dioxide and water. We make carbon dioxide during this stage, and this is when water is made. It occurs in the cristae, the inner mitochondrial membrane. The amount of ATP varies, but it's on average between 28 to 32 molecules of ATP. And then our loads of our reduced carriers change too, depending upon what's happening in the environment. So if we are to add up the total of ATP, it ranges from 32 to 36 molecules of ATP for every 1 glucose. This is just stage 3, right? You might want to add a total column into your table where you sum everything across each of these rows. So when I ask you on an exam or a quiz, the total yield of ATP per 1 glucose, as we go through all stages of aerobic cellular respiration, the answer would be 32 to 36. Right? You're going to give me a range because it produces a range depending on the circumstances. I'm trying to give you very clear guidance because this can be a little confusing, and I don't want you to be figuring out, okay, well, is it 34? Is it 35? No, the answer is a total of 32 to 36 molecules of ATP for every 1 glucose, providing all 3 stages of aerobic cellular respiration has been completed. So going back to the figure we started at, we can now add in our quantities of everything and see how the electron transport chain is generated and feeds into the production of energy through oxidative phosphorylation. Yes, we have the chain, but we also have this process of chemiosmosis, which is where the majority of ATP is produced. So we make 32 to 36 molecules of ATP per 1 glucose. So I'd encourage you to take the version of this figure that is at the beginning of the slide set and try to fill it in yourself with as much information as possible, or try to draw it yourself. All right, we've got 2 more topics we need to cover before we wrap up this module. The first is the anaerobic pathway. And then the last topic is we're just going to put it all together. So fermentation is anaerobic respiration. And this means that that energy is being made without utilizing oxygen. It doesn't necessarily mean that there's no oxygen in the environment. That's different. We are not using oxygen in the chemical reactions of fermentation. So it's a way to harness energy without requiring oxygen in the chemical reaction. Now, fermentation does use glycolysis. So stage 1 glycolysis is utilized in both aerobic and anaerobic cellular respiration, otherwise known as fermentation. So we do produce 2 molecules of ATP, a net production of 2 ATP for every 1 glucose. Glycolysis is glycolysis. Very importantly, though, we have to deal with our reduced shuttles. Part of the process of glycolysis is to take the oxidized NAD+ and reduce it to NADH. That's still the same. What is different between fermentation, this anaerobic pathway, and just glycolysis is fermentation has the pathway to oxidize those carriers so they can be used again. We cannot load any more electrons onto NADH. There's only a site for 2. So both of those have to come out through oxidation in order to use the carrier again. So one of the fundamental problems with anaerobic respiration is that the reaction would stop after producing those net production of 2 unless we had a way without being able to oxidize those carriers. So our body does this through what is called lactic acid fermentation. There are many different fermentation pathways. We use lactic acid fermentation. Bacteria and yeast can use alcohol fermentation or ethanol fermentation. So the products are slightly different, but the process is the same. Importantly, the NADH is oxidized back to NAD+, which means it can be used again to process another molecule of glucose. Pyruvate is reduced into lactate. Okay, and that burns. You've probably all felt lactic acid fermentation after running a distance or sprinting. You know that burn, that sting you get in your muscles? That is a buildup of lactic acid, and it is acidic. Fermentation always produces an acid and a gas. Those are two products that are made for every fermentation reaction, acid and gas, as well as carbon dioxide. So that acid is actually what's burning our muscles, but we need it. We actually need this anaerobic pathway, even though we are aerobic organisms. The value of the anaerobic pathway for us is that it gives us a turbo boost when we need it. This is what gives our body the ability to sprint as opposed to run just a steady distance. So it's a very important process in us too. I don't want you to just associate it with, with making yogurt or beer or cheese. We use it too. So here's the pathway. This part is glycolysis. You've seen this now. It begins with a 6-carbon glucose. The 6-carbon glucose is broken down into 2 molecules of pyruvate, each containing 3 carbons. A net production of 2 ATP are made through substrate level phosphorylation. 4 are made, 2 are used. So that leaves us with 2. 2 molecules of NAD+ are reduced to 2 NADHs. That is the end of glycolysis. What fermentation does is it takes pyruvate and converts it to lactate, and doing this oxidizes the shuttle. So now we've taken our reduced shuttle and removed the electrons out of it. Oxidation is loss. And now this NAD+ molecule can be utilized to process another glucose. So that is the key difference between glycolysis and fermentation. Glycolysis produces a net production of 2 ATP. It produces 2 pyruvate. That's what glycolysis does. What fermentation does is it oxidizes the carriers so they can be used again. Without fermentation, the reactions would stop and only a total of 2 ATP would be produced, period. Instead, with fermentation, we can make a total of net production of 2 ATP for every molecule of glucose that is processed. So when I play the video at the end of this slide set, please pay attention to what Mr. Anderson explains about fermentation. I think he does a really good job explaining that in his video. In fact, it was one of the videos I watched that, that helped me to really understand the process. Now let's talk about anaerobic, what that means. There are organisms that are anaerobic organisms. Obligate anaerobes are poisoned by oxygen. They can't live in the presence of it at all. It's a toxin for them. And there are certain bacteria, like the bacteria that causes tetanus, Clostridium, that is an obligate anaerobe. So they're literally poisoned by oxygen. We tend to find the obligate anaerobes in soil or ponds that have little circulation. That's where they exist naturally. There are also facultative anaerobes. Facultative anaerobes are not poisoned by oxygen. They can live with it in the environment. They just don't utilize it in the chemical reactions of metabolism. So facultative anaerobes are able to make ATP really either way. There are many facultative, or there are bacteria that are facultative that can switch metabolism. Basically, if oxygen's present, they're going to utilize oxygen because it's much more efficient in producing 32 to 36 ATP per 1 glucose, whereas anaerobic would only make 2 ATP per 1 glucose. So a facultative organism really has the best of both worlds, where it can metabolize depending upon the specific environmental conditions at the time. But importantly, facultative anaerobes are not poisoned by oxygen. So many yeast and bacteria fall into this category. Okay, so wrapping it up, and then we'll take a look at that video. Putting it together. So we've been talking about, we began the semester by learning about the chemistry. We learned about different molecules and how they're made into bigger molecules, for instance, how a polysaccharide is made. But now we're going to look at it a little bit differently. So cellular respiration is beginning with those big molecules and breaking them down so that we can extract and harness the energy out of it. So we can get energy, we can produce energy from sugar, yes, but we can also produce it from fat and from proteins. In fact, fats produce more energy than do carbohydrates or proteins. The problem with America is that we have an obesity epidemic, meaning that there is too much obesity. So we kind of get this connotation that fat is bad, but it actually is bad when there's too much of it. Fat serves a purpose, right? We've evolved mechanisms to deal with it. We need it, but we have to have the right quantity so that we don't develop cardiovascular disease. So fats do make great cellular fuel. Why? Well, they have so many hydrogens in those tails. So that means they have lots of energy. They have lots of electrons that can be shuttled into the electron transport chain. So they can make a lot of energy. In fact, if we begin with 1 gram of fat, 1 gram of protein, and 1 gram of carbohydrate, fats make twice as much ATP per same quantity than do a carbohydrate. So fats are more efficient at producing ATP. But proteins too can be used that way. Our body, though, is going to preferentially burn sugars and fats before digesting as proteins, right? An indication that a person is suffering from malnutrition occurs when we start to see the body using muscles as an energy source, using the proteins in that muscle. And that does happen when the body needs it. It's going to take it. So we begin our whole process of digestion by consuming the macromolecules that make up our food. Let's look at this a little bit differently. This is not new information to you now. It's just being presented differently. The foods can be broken down into carbohydrates, fats, and proteins. Sure, we might be eating some nucleic acids too from bacteria or something, but the gut environment gets rid of them. Carbohydrates are broken down into their monosaccharides, which then can feed into glycolysis. Fats are made of their glycerol head and their fatty tails. Remember that fatty acids have 3 fatty tails. Phospholipids have 2. Fatty acids have 3. The glycerol part of a fat can be converted into an intermediate G3P, which is then converted into pyruvate. So that can feed into the citric acid cycle, right? Pyruvate is pyruvate. The fatty acid tails, they are harnessed or metabolized beginning in the transition step between glycolysis and citric acid. Proteins are made of amino acids, and it does depend upon which amino acid we're talking about. So some amino acids are converted to pyruvate, some are converted to acetyl-CoA, and some enter the citric acid cycle directly. Everything in the citric acid cycle feeds in to oxidative phosphorylation, providing oxygen is present in the environment. And all of these pathways of cellular respiration generate energy. That is the purpose of cellular respiration. It is to take the macromolecules we eat, break them down into their building blocks. And that way our cells can use those building blocks to make whatever it needs. But also, very importantly, the cell is able to generate energy in the form of ATP that may be saved and utilized for other needs, whatever the cell needs. So our food really those are the raw building blocks that are needed so that our cells can make anything it needs. So cells utilize these building blocks to make its own structures, but also to perform the various functions that each cell needs. So it is our food that really is that source of raw building blocks. So putting it all together, putting these pathways all together and summarizing metabolism, we just looked at this component of it, but ATP is the energy that supplies the synthesis of proteins and fats and carbohydrates that our cells are able to build by digesting the food molecules that we eat. So again, fats can be broken down, can be digested into glycerol and the fatty acids. The fatty acids are processed into acetyl-CoA, where the glycerol is processed into a molecule that can go through glycolysis. So our body takes what it needs and breaks it down into the forms that we can utilize, that then our cells and tissues are built out of. And of course, this takes energy to make these molecules. So these are the study slides. Please make sure you are intimately familiar with these slides, backwards and forwards. These you've seen within this module. But I put these back here as a way to emphasize to you that these are important concepts to understand for exam preparation. So you've got that, and here's your fill-in question from chapter 6. The answers for these are found in your textbook. Okay, now let me switch over. Excuse me, it's just going to take me a brief moment to switch over to the video that I'd like to play for you. Let me just get that up, and that will wrap up module 10. Just a second, sorry, it's taking me a little bit to navigate here. So this video is about, it's about 15 minutes long. Let me go ahead and share the screen with you so you can see where to find it. So this is Canvas. It may look a little bit different to you because I have it in the instructor view. But if you come down to the Mod 10 supplemental YouTube videos, this is where you'll find these 3 videos. So I'm going to play the second one for you. I need to switch the shares on that so that you can see it. But please do look at all of those videos. They're there because they're worthwhile to view. I don't put anything up here that is wasteful.
It's Mr. Anderson. In this podcast, I'm going to talk about cellular respiration. Sometimes students confuse respiration, breathing, just breathing in, and cellular respiration, and they are linked. Cellular respiration, however, is going to take place at the level of the cell, more specifically inside the mitochondria. And so we need oxygen for that. But it's basically taking our food and then breaking it down in the presence of oxygen to make ATP out of it. What if you're a bacteria? Can you do respiration? You sure can. You don't need a mitochondria to do respiration. They can actually use their outer membranes to do aerobic respiration. And so basically, if you are a track athlete, so this is Usain Bolt right here, when you run, you're using respiration to make energy in the form of ATP and allow your muscles to move. And so this is a quick little study I did. I took all of the world records currently right now from the 100 to the 10,000. And so this is Bekele is the guy who owns the 10,000 meter record. And what I did is I figured out their pace. In other words, how fast they're running. This would be in meters per second for the 100, the 200, all the way down to the 10,000 meter run, which is a little over 6 miles. And what you can see in this graph is that the pace is quickly going to drop off, and then it's pretty much going to stabilize. And if we were to go out to the marathon or continue, basically, we're pretty effective at running at a specific pace, but we die really quickly when we're sprinting. And so a way to think about that is the two things we're going to talk about in this podcast is we're going to talk about aerobic respiration. So aerobic respiration is going to be respiration in the presence of oxygen, but we also have almost like a turbo button that is anaerobic respiration. And so if we really need to go fast, we can get that extra speed that we have up here by doing our anaerobic respiration. The problem, if you've ever run, is that when you get out to this 400 meter, you get a buildup of lactic acid, and it's incredibly painful. And so you can't keep that pace going. Example of a lab I did in class was, and it's weird how this exactly mirrors it. What we did was the muscle fatigue lab. So basically you had a tennis ball, and in one hand, you had to squeeze it as many times as you could in 10 seconds, and then do another 10 seconds and do another 10 seconds. And this is the class average. So the class average was, it looks like around 25 times in 10 seconds. But you can see that it quickly drops off and then it kind of levels off. And so the same thing, this would be that aerobic respiration. And then this is going to be that anaerobic respiration. And it was fun to see the students' faces, because as they start to go anaerobic, your arm just starts to build up at the lactic acid on the inside of it. But before we get there, let's talk about respiration and what it's for. It's for heterotrophs. So we're heterotrophs. And basically what we're doing is we're taking organic compounds in the presence of oxygen, and we're converting that to carbon dioxide and water. What else are we generating? ATP. Now, what kind of things are doing this? Animals, fungi, bacteria are all heterotrophs, and they're using the organic material to actually make energy. Luckily, we have autotrophs like plants and algae, and basically what they're doing is they're converting that carbon dioxide and water back into organic materials. The only thing that's a little deceptive is that plants are also going to break down those organic compounds, and so they do cellular respiration as well. And so everything's doing cellular respiration. It's how we get energy out of our food. Okay, here's our equation. And again, if you know what photosynthesis is, this is simply the opposite. We're going to take glucose in the presence of oxygen. So here's glucose and here's O2. And then we're going to break that into carbon dioxide, water, and then we're going to generate a little bit of ATP. Now, where is the energy sit? The energy sits right here in this hydrogen on the outside of that glucose. And watch what happens to that hydrogen. It's going to fall down and it's going to grab onto the oxygen, because oxygen wants electrons. And so that's where the energy is coming from. And what the energy is used to do is it's used to make ATP. And ATP is that little fuel that we use in all of our cells. This slide is funny, but it's saying this: behold the power of oxygen. So this fire comes from oxygen pulling electrons close to it. And so there's a huge amount of energy found inside that pull of electrons towards oxygen. Now, if we were to do this inside our body, we could get a lot of energy out of our food, but we would also burst into flames. And so we do it in a really controlled process. Just like when we learned photosynthesis and we had to learn the parts of the chloroplast, when you're learning the cellular respiration, you have to learn a few parts of the mitochondria. So first of all, we have these folds on the inside of the mitochondria. Those are called the cristae. And basically what we have is two membranes. We're going to have an inner membrane right here, and then we're going to have an outer membrane right here. And then this space in the middle is called the inner membrane space. And on the inside, mitochondria, we think, used to be bacteria of their own. And so they'll reproduce through binary fission. They have their own DNA, they have their ribosomes, but they are kind of almost living inside us, not as a parasite, but as a symbiont. They're actually helping us as we generate energy. So there are 3 steps in cellular respiration. Let's start with the first one. So the first one is going to be glycolysis. The second one, normally we think of as the Krebs cycle. And then the third one is going to be the electron transport chain. And so the first one, I love this diagram here because it's putting glycolysis outside the mitochondria. And so this is going to take place outside the mitochondria. Where would that be? Well, that would be in the cytoplasm of a cell inside your body, or it would be right outside of bacteria. But what happens in glycolysis? Basically, we're taking glucose. Glucose is a 6-carbon molecule. And in glycolysis, we're going to break that down into 2 molecules of pyruvate. Each of those have 3 carbons inside it. So the 2 3-carbon molecule, that's what glycolysis does. What do we generate in there? Well, we generate a little bit of ATP. For 1 glucose molecule in glycolysis, we're going to make 2 ATP. The other thing that we make is a chemical called NAD. What we're basically doing is we're transferring high energy electrons to NAD, and we're adding protons to it as well. And we'll get to NAD in just a little bit. Let's follow pyruvate then. Pyruvate is going to diffuse into the mitochondria, and then we're going to have this pyruvate dehydrogenase complex. And basically what it's going to do is it's going to convert that 3-carbon molecule into acetyl-CoA. This is coenzyme A. So basically now we have a 2-carbon molecule that is going to go into the Krebs cycle. Now, since we're going from a 3-carbon pyruvate to a 2-carbon acetyl-CoA, we're giving off carbon. And that carbon is going to be given off in the form of carbon dioxide. And so when you breathe out, a third of that carbon coming out of you is going to come right here from this complex inside the matrix, we call this. I should have said that before. This is the matrix on the inside of the mitochondria. Okay, let's keep watching acetyl-CoA. So it's a 2-carbon molecule. Where does it go next? It goes to the Krebs cycle. And so in the Krebs cycle, we're going to break it down further, and we're going to get rid of these 2 carbons in acetyl-CoA, and we're going to give those off as carbon dioxide. So we're getting rid of carbon dioxide. What else are we producing in the Krebs cycle? You can see here that we're producing a little bit of ATP, 2 ATP, but we're also adding energy again to NAD. And we're adding energy to its friend, we'll call this FAD. And so what do NAD and FAD have? They have these high energy electrons, and they're going to carry those electrons to the 3rd step, which is the electron transport chain. Okay, let's get to the electron transport chain then. All of our energy pretty much that was in glucose is now in NAD and FAD. So they're going to transfer their electrons, and those electrons are going to go through what's called an electron transport chain. Basically, they're moving through a series of proteins, and the energy of those proteins is used to pump protons. Protons are going to be hydrogen ions to the outside of this inner membrane into what's called the inner membrane space. So now we've built up all of these protons right here. What happens to the electron? The electron is going to be added to other protons and oxygen that we breathe in, and that's going to make our byproduct, which is going to be water. And so let's slow that for just a second. The oxygen that you breathe is moving in here, and it's going to be that last electron acceptor right in here in the matrix, and we're going to take the protons. What happened to those protons? They'll actually flow through a protein called ATP synthase. Those protons will combine with the electrons and the oxygen, and it's going to make water, which is going to be a byproduct of that. Now, how much ATP do we make down here? Well, we can make around 32 or 34 ATP in this last step. And so in the electron transport chain, we're making a whole heck of a lot of energy. And so it's worth taking a look at how that actually works. So let's go to the electron transport chain. So kind of to situate ourselves, what do we have? Well, we have NAD, our friends NAD and FAD. What are NAD and FAD passing off? They're electrons. Those electrons are going to move through the electron transport chain like this. Every time they go through one of these proteins, it's going to pump another proton ion out, because that's the other thing that NAD and FAD are bringing. They're bringing these hydrogen ions. So we're going to pump these ions out here, and pretty soon what you get is a heck of a lot of positive charge out here in this inner membrane space. There's no place for it to go. In other words, every NAD that we drop off, we're going to move these electrons down, And we're going to generate a whole heck of a lot of positive charge in this intermembrane space. Now, if you look right here, fat is actually dropping its electron a little bit farther down, so it can't generate as much, but we're pushing out either 3 protons or 2 protons, depending on if it's NAD or fat. Okay, what happens to all of these protons out here? They can't go anywhere. They can't go outside the mitochondria. They can't come inside the matrix, but they can move through this. This is called ATP synthase. That's the name of this protein right here. And basically, this is the site of ATP synthesis. And so basically, as every proton flows through it, every proton that comes through, we're going to generate ATP. And it almost works like a little rotor that every time a proton goes through, it switches it and it attaches that phosphate onto ADP to make ATP. And that's why in the electron transport chain, we can make all of that ATP. There's nothing special about it. It's just that we're storing all that energy. And instead of releasing it in a ball of fire, we're releasing it in little bits to make a heck of a lot of HTP or ATP. Okay, so there's a problem. What happens if you don't have oxygen pulling that electron the whole way? Or let's say you don't have mitochondria present. Well, then you have a problem. And the problem is this. It's okay to take glucose during glycolysis and break it into two pyruvates because you're going to make a little bit of ATP. But the problem is that you're adding those electrons to NAD. And so basically what's happening is that we're adding electrons to NAD plus, and we're transferring it to NADH. And so pretty much what happens is there's no more of this. And so glycolysis has to shut down, even though we can make a little bit of ATP with each breakdown of glucose. Eventually there's no NADP plus, and so the whole process has to stop. And so nature, of course, has a solution to this. And the first one is called lactic acid fermentation. This takes place in your muscles, especially when your muscles are under a huge amount of stress, like if you're sprinting or if you're holding your breath for a long period of time. And so basically what's going on, again, there's no oxygen, there's no mitochondria. So let's look what happens. Basically, your cells are taking glucose in glycolysis and breaking it down into 2 pyruvate molecules. So we were stuck here, remember, with the NADH. But then there's a further conversion. Basically what you're doing is you're converting that pyruvate down into lactate or lactic acid. The nice thing about that is it's accepting these electrons so we can make more of this NAD plus, and then this can be recycled again. And so basically what happens is that you can have this process occurring with glucose, over and over and over and over and over again. And every time you do that over and over and over and over again, basically you're making 2 ATP each time. And so if you've ever done sprinting, when you're sprinting, you're getting aerobic respiration, but you're also doing anaerobic respiration on top of that. The problem with that is you're going to build up lactate in your muscles. And that lactate is like a toxin. You're going to have to break it down. And that takes oxygen. And so if you've ever watched a sprinter, especially somebody who's run like the 400-meter dash, when they're done and they're interviewing them, they have a hard time doing an interview because they have to keep breathing to take in more O2 and eventually get rid of that lactate. And so lactic acid fermentation is going to take place in some bacteria and in muscle cells, but we have another solution to this in bacteria, anaerobic problem of stopping right here with this full NADH, and that's called alcoholic fermentation. Alcoholic fermentation works the same way. Basically, we break it down into pyruvate, and then we break that further down into a chemical called ethyl alcohol or ethanol. It's accepting these electrons so it can recycle this NADP plus again. The only difference here is that when we made lactate, that was a 3-carbon molecule. When we do alcoholic fermentation, what we're making is carbon dioxide, and we're giving that off. And so if you were to take yeast and put them in a bottle with a bunch of fruit juice, basically what they'll do is they'll use up all the oxygen, then they'll switch to alcoholic fermentation. What are they going to build up? They're going to build up ethyl alcohol. That's simply how we make wine. And there's also going to build up carbon dioxide, which we could let go. Or if we're making beer, that's the carbonation that we're going to find in beer. And so again, cellular respiration is just a quick way to get energy out of glucose. We use glucose as an example, but we can do cellular respiration on pretty much any type of food. And it's a way that we get energy. And we're doing it, bacteria are doing it, plants are doing it. And I hope that's helpful.
All right, class, that completes the video there. And also, sorry, it's going to start talking again. And also the lecture for Module 10. So I hope that this helps to answer some of your questions. Please reach out with any questions by email or come to office hours. You do have a quiz later this week. Usually your quizzes would be on Tuesdays, but so you have some time to study. Your quizzes later this week. Take care, everybody. I hope you're doing well. Bye-bye.